or my greetings to all of you in whatever region you are. So thank you so much, organizers, for this opportunity. And as title suggests, I will talk about arginine citrullination of RNA polymerase II controls transcription regulation in breast cancers. And so first of all, I will start with the fact that what is this arginine citrullination? So arginine citrullination is a post-translational modifications, and as you can see over here, in which the peptidyl arginine is converted to the peptidyl citrulline with the help of the enzymes, or the family of the enzymes is called PADI, which stands for peptidyl arginine deaminases. And as you can see over here, that these enzymes require calciums to maintain its catalytic activity. But much more importantly, that still now we don't know whether this reaction is reversible. So this is a unidirectional reactions, and this positively charged amino acid modified to non-coded amino acid, which is citrulline. And it's always a question that, of course, so many post-translational modification have very important role, but this is something which I would be speaking in next 15-20 minutes, that this non-coded amino acid have a more carcinogenic effect in breast cancers, and it may be in several other cancers. So the next question is, this is just to highlight you that in humans, there are 5 different PADI members, and these all are present in one common locus on chromosome 1. And surprisingly, this whole PADI locus is a part of the one of the topological associated domain, which this suggests that this might be, there is a evolution of this PADI family members in higher organisms, including humans, and that's one of the reason it might be evolved to regulate the transcription regulation. So the next and very important point is this, that what is the function of this citrullination? So during citrullination, first thing what we know so far is happens that there is a reduction in hydrogen bonding, which affects the interactions between cohistones, histone H1 with the DNA, and that leads to the chromatin decondensation. So much chromatin decondensation and transcription activation might be increased in this region. And much more importantly, which you can see over here, is this, that whenever there is a citrullination takes place in any proteins, that might, because there is an increase in the hydrophobicity of the proteins, and because of this, the protein-protein interactions could be affected. And as per means what you can imagine, it could affect in both manner, like positively and negatively. But this is something we can understand, that if interactions of the two proteins affected in a one specific complex, it can lead to different kind of pathophysiological conditions. And probably this explains that why does the elevated level of citrullination found to associated with several pathological conditions, including the breast cancers, which will be the topic of our discussion. So this work I have done in my previous lab, which is this exciting atmosphere in the Center for Genomic Regulation. And I would like to mention that in this study, we have used the model system as a breast cancer cell line. So we use the epithelial-derived breast cancer cell lines. So here I will present you some data set which we have done, set up the experiment in epithelial-derived T470 cells. So as I show you before, out of the 5 human PADI family members, these cells only express PADI2 and PADI3. So this is quite unique in this sense. And most importantly, we focused on the PADI2, and the reason was that among 5 PADI family members, PADI2 is the one which is highly overexpressed in tumor patient samples in comparison to the healthy control individuals. Suggesting, so the whole idea of this work was that we wanted to understand what could be one of the molecular mechanism why which this enzymes is so much overexpressed and might have some pathological functions. So in this regard, we started to understand the transcription regulation affected by, or regulated by these enzymes. So here I'm presenting you the RNA sequencing data. As you can see is a Volcano plot. X-axis showing you the log to fold change and y-axis showing you log 10 transform value. And this is done after PADI2 knockdown. So only thing you can have a notice over there that actually PADI2 knockdown drastically affects the transcription regulation. We can see that approximately 2000 genes are downregulated significantly, and more or less the same number of genes are upregulated, which might be the effect that once you have the downregulation, that it also inversely or signaling affects the upregulation also. So we wanted to understand that how specific this effect is. So we start doing the validation, and I'm showing you here the validation part. So the first thing you can see over here, because as I mentioned, these cells express PADI2 and PADI3. So we did knockdown of PADI2 and PADI3, and the PADI2 dependent genes is only shows reduced in expression after PADI2 knockdown, but not after the PADI3 knockdown, suggesting that this is very much likely is a PADI2 specific actions. And given the biological relevance of this set of the gene, we decided to perform some assays. And one of the critical assays, we wanted to know whether this enzyme is responsible to maintain the cellular proliferations. So we did the cellular proliferation by one of the very typical bromodioxyuridine incorporation. And you can see over here that the PADI2 knockdown as well as the inhibiting PADI activity is significantly reduce the cellular proliferation, suggesting that PADI2 is required to maintain the gene expression and cell proliferation of breast cancer cells, which is quite good to know. But the next question is this: what is the molecular mechanism behind this? Means where is PADI2 is targeting in the genome or in breast cancer cells? So as I already said, so the question was that how does PADI2 mediated citrullination could affect the transcription regulation? And to understand these questions, we applied several approaches, and one of the approach we did very hardcore way to understand this mechanism. We try to figure it out what are the proteins which is interacting specifically with the PADI2 in the nucleus of these cells. So we have used the immunoprecipitation. So we use the PADI2 specific antibody and followed by the mass spectroscopy using peptide identification. We found very nice data that more than 200 unique protein, which is part of the transcription machinery. But I would like to highlight one of them, which is master regulator of RNA transcription machinery and is the RNA polymerase II. So I'm showing you here the validation part of this global proteomic data. So you can see over here, this is the PADI2 immunoprecipitation. You see PADI2, but you also see active or the phosphorylated form of RNA polymerase II. Some of you already know that the polymerase can be phosphorylated at the 5 position and the 2 position, and we can figure it out. PADI2 interacts with serine 5 as well as serine 2 phosphorylated form of RNA polymerase. In simple terms, it means that the active or elongating form of RNA polymerase interacting with the PADI2. Not only this, you also see over here that PADI2 interacting with the components, which is binds with the active RNA polymerase II, and one of this component is made up of CDK9 and the cyclin T1, which is known as the positive transcription elongation factor B complex. So this is all right that the PADI2 interacts with the RNA polymerase II. But just to make whole story short, I would like to have your attention on the fact is this is just a cartoon of RNA polymerase II. So in human, the RNA polymerase II have the very long this carboxy terminal domain, and this carboxy terminal domain is a low complexity domain. And in human, actually, we have 52 heptapeptide repeats, which is present on this long carboxy terminal domain of RNA polymerase II. So the dots is showing you 52 repeats. And if you have paid attention, the red dots is showing you the one which is having this repetitive sequence, consensus sequence. And the green dots are the one which is not following this consensus sequence. And among these green dots, there are two repeats which is actually have the arginine. So one in the repeat number 2 and the another one in the repeat number 3. Just to make whole story very short and finalize that, we found that PADI2 actually catalyze the citrullination of this specific arginine, which is kind of in between or the heart of the CTG domain. And we also demonstrate that this citrullinated version of RNA polymerase II is present in the cells. So here I am showing you the immunostaining, and this is the triple labeling immunostaining, meaning that in the red cells, you are seeing the citrullinated form of RNA polymerase. Blue here is representing you the label of PADI2, and the green is representing you the elongating or the active RNA polymerase II. You can see they are quite localized very well, as you see the red spot. On top of that, you can see that they are co-localized as quantitation has been done more than 100 cells. So this suggests that the PADI2 mediated or PADI2 catalyzed citrullinated form of RNA polymerase is most likely like an integral part of the transcription machinery. But the question is that why RNA polymerase need to get citrullinated? Means what is the functional consequence if we don't allow citrullination to happen? What could happen in the cells? So I will present you a couple of slides for this part now on. So as I already mentioned, that why RNA polymerase need to undergo citrullination, and specifically on this specific arginine. And I think I will try to convince you by showing this data. So we engineered the system in the lab where we use these cells and we express two kind of RNA polymerase. In the one cells, we express normal wild type RNA polymerase, and the another cells, we express the citrullinated, means the RNA polymerase which could not be citrullinated. And we decided to understand this effect on the transcription regulation. So here again, I'm showing you the volcano plot, and you can see that when the RNA polymerase II could not citrulinate, it drastically leads to the down regulation of many thousands of the gene, as you see the blue section of the volcano plot. Whereas the upregulation of gene is not so evident, it's just 150 in comparison to 1884, suggesting that this citrullination mark on RNA polymerase is kind of a positive regulator to allow transcription to happen. You are also seeing over here some of the genome browser snapshots. So you can see when you don't allow, when you do the knockdown of PADI, you deplete the transcription. But when you don't use the mutant form of RNA polymerase, which could not citrulinate, you also reduce the transcription over here, as you see over here. And the another biological context that why does citrulination is required, as I showed you, that if you do the knockdown of PADI, cellular proliferation is not go very well or not very appropriated. This is what also happens when you don't allow polymerase to citrulinate. And the main reason for it is that because cells are arrested at the G1 phase of the cell cycle. So you are seeing over here just a snapshot, and here is the quantitation. So this is quite okay that the RNA PADI2 is the one which is very highly expressed in breast cancer patients and the breast cancer cells, citrulinate specific arginine of RNA polymerase. And this citrullination mark most likely is the driver of the transcription and also the cellular proliferation, to maintain the cellular proliferation. But one more link was missing over there. As I mentioned in the initial slides of my talk, that most likely the citrulination, when this happens, this modulate the protein-protein interaction. So I was very wondered that, okay, the RNA polymerase citrulinated, but how does it could affect the protein-protein interactions, or which proteins interactions might be affected in this case? Because we know RNA polymerase is a huge machinery and have a so much protein, is a huge protein complex itself over there. Again, I will just try to make my talk very clear to you. So I want to share with you that what could happen to the RNA polymerase if we don't allow PADI2 to citrullinate the RNA polymerase II. And the answer is over here. But before we understand the answer of my questions, I would like to have your attention on this specific cartoon. So what happened in the humans, that in human, more than 70% of the genes actually start transcription, but shortly after the transcription initiation, the RNA polymerase post or just accumulate, just stop for a while. And at this specific moment, it requires a green signal, meaning that it requires the proper recruitment of some of the protein complexes. And once this protein complexes is loaded, this is a green signal, and then the transcription goes very well, or polymerase elongation goes very well. So here I represent you just very one complex, which is the critical complex, and this is PTFB, positive transcription elongation factor B complex. And we know, and I also present you that this complex also binds with PADI2. So what happens normally, PTFB is there, and once it's recruited properly to the RNA polymerase, the transcription goes on. So I was trying to answer your questions that what happened to the RNA polymerase if we don't allow citrulination to happen. So we use these engineered cell lines, and then we perform the chromatin immunoprecipitation of RNA polymerase II. So chromatin immunoprecipitation followed by the sequencing. And the data is over here. So the mutant version of the polymerase, which could not citrulinate, actually leads to accumulation of RNA polymerase near the transcription start site, as you can see here in the red area of the plot. And the wild type version of RNA polymerase, which could citrulinate, is the recruitment over the transcription start site. So in other words, the mutant version of RNA polymerase, which could not citrulinate, actually is stuck in this kind of the situation. It just posed. It could not enter into the next phase of the transcription elongation. You also see here the quantitation of the whole posing index, and it's shown over here that the mutant version have a more posing index than the wild type, meaning that this simple terms is that if you don't allow polymerase to happen, polymerase to citrulinate, it just get stuck at this specific region and don't enter into the productive phase of the transcription elongation. So just to make a final conclusion to what I presented to you till now is that I presented to you that in RNA polymerase, there is a very specific arginine, which is kind of the hot spot for many reasons, many biological reasons, is specifically citrullinated by the PADI2 among family members. And this PADI2 and this citrulination mark actually is a sensor for PTFB complex, the positive transcription elongation factor B complex, for the proper recruitment. So once the arginine is citrulinated, it facilitates the interactions of the PTFB. So this go this way. And this is how it allows polymerase to enter into the next phase of the transcription elongations. So it's like a switch or is a sensor to facilitate more transcription, more cellular proliferations. And why it makes sense in the breast cancer biology is that, as I showed you, that the PADI2 is the one which is highly expressed among PADI family members, and it was not known what could be the pathological relevance, and overexpression of this PADI actually associated with the poor prognosis. So this is one of the therapeutic marker, of course, need to be investigated. And with this mark, I would like to stop over here. I would like to thank you, all of you, for your nice attention. And I would like to thank my previous lab at the Center for Genomic Regulation in the lab of Miguel Vieto, where I performed this whole set of the experiments. And of course, my new affiliations at the Montpellier Cancer Research Center and the INSAMS. And thank you so much for all your attention. I'm all up to have any questions.
Great. Thank you very much, Dr. Priyanka.